So this presentation is all about how we know that DNA is the genetic material, and we're going to take a chronological look through history determining that DNA is the genetic material. One of the first experiments conducted to lead us to the conclusion that DNA is the genetic material was performed by Frederick Griffith in 1928. He was trying to create a vaccine against pneumonia, and so he was dealing with two different strains of the pneumonia bacteria, one of which had a smooth surface around the cell membrane, so that was named the S strain, and the other one had a rough surface, so that was the R strain. Griffith did a series of 4 different groups taking a look at these series of pneumonia bacteria. The first group was the S strain, and when he injected that into mice, he found that the mice died. The second group had the R strain, and when he injected those into mice, he found that the mice lived. The third group, he took the S strain and he killed the bacteria by heating it up to a temperature at which it can't live, and he saw that when he injected those heat-killed S bacteria into the mice, they actually lived. The fourth group, he took the R strain which had lived, and he had those living bacteria, and he mixed them with the heat-killed S strain. And when he mixed those together and injected them into the mice, he now saw that those mice died. So the DNA from the S must have somehow gotten into the R cells, causing the R strain to become S strain, and the S strain we know is deadly. This picture just demonstrates the 4 groups. The first group we have here is that we inject the S and it dies. The second group was we inject the R and it lives. The third group was the heat-killed S, and when we put those in the mice, they lived. So when we combine the bacteria from these two and put it in another mouse, that mouse died of pneumonia. And when they looked at the bacteria, they found not R, nope, that wasn't it. When they looked, what they found was actually S. So Griffith made the conclusion that something from the S strain converted the R strain into the S strain. In 1944, there were 3 scientists that conducted another experiment that helped lead to the conclusion that DNA is the genetic material. This experiment was conducted by Oswald Avery, Colin MacLeod, and Macklin McCarthy. They used that same heat-killed S strain of pneumonia that we saw in the last experiment with Griffith, and they removed the carbohydrates and the lipids, and they were able to split the sample into 3 groups. Here you can see in this diagram, this was the heat-killed S. They separated out the lipids and the carbohydrates. So to this one, they add proteinases. So that's going to kill all the proteins. And then to this one, they added ribonuclease. And what that does is this sample has no RNA in it. And the final sample, they add DNA nucleases, and that's going to remove all of the DNA. So this one has no protein, this one has no RNA, and this one has no DNA. They then decided to take those 3 test tubes on the previous slide, and they added heat-killed S. pneumonia cells to each of those test tubes. They took samples from each and injected them into mice. What they found is that the only mice that survived were the ones where the DNA nucleases had been added. So from the test tube where there was no DNA. From that, they were able to conclude that something about the DNA must have been what was killing the mice. In 1950, Chargaff did his work, where he was taking a look at DNA samples. And in every sample, he noticed that there were equal amounts of adenine and thymine, and there were equal amounts of cytosine and guanine. So Chargaff is credited with coming to the conclusion that adenine must pair with thymine, and cytosine must pair with guanine. In 1952, 2 scientists, Alfred Hershey and Martha Chase, were the ones really kind of definitively put that nail in the coffin that DNA must be the genetic material. There were still a lot of skeptics out there, but they were able to prove beyond a shadow of a doubt. So let's take a look at what they did. They utilized a virus, And viruses are really simple. They have a protein coat on the outside and they have DNA inside. So at this point, the debate was really, is protein the genetic material or is DNA the genetic material? So viruses work by attaching to cells and they inject whatever their genetic material is into the cells. So Hershey and Chase were using this to their advantage. They were able to label the protein coat on the virus with radioactive sulfur because sulfur is found in protein, but it's not found in DNA. They then infected bacteria with this virus. They separated the bacteria and the protein coat, and they looked for where they found the label. Was the label going to be inside the cell or outside the cell? And then they did the exact opposite. They were able to use radioactive phosphorus to label the DNA because we find phosphorus in DNA, but not necessarily in protein. So they then used this DNA that was heavy with phosphorus and it was inside the virus and they infected bacteria with this virus. Again, they separated the bacteria from the protein coat and they looked for where they found the label. Was the label going to be inside the bacteria or was it going to be outside? Da, 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 da. The results of the Hershey-Chase experiment show that here we have radioactive phosphorus. So here's the DNA. When they looked for the label after injecting the virus, the label was found inside the cell. They didn't find any label outside the cell. When they did the same thing with the radioactive sulfur for the protein coat stayed outside and we found a label outside. So what was getting into the cell had to be DNA. And this was determined to be the definitive answer. This is where all that research stopped. We now know DNA is in fact the genetic material. When they found the label inside the bacteria with the DNA having been labeled, they made the conclusion that DNA must be the genetic material. At this point, any controversy over whether it was DNA or whether it was protein that was the genetic material, that controversy stopped because this was definitive proof. DNA is the genetic material. Now we go to Franklin and Wilkins. So Rosalind Franklin was working in the early 1950s and her supervisor in the lab was Maurice Wilkins. And Franklin found a way to take x-rays of DNA and it created kind of a photograph of the DNA. And then they were able to use those pictures of the DNA to indicate that DNA was a double helix because at this point we still didn't know much about the structure of DNA. So while Franklin figured that out, Wilkins shared that information with Watson and Crick in 1953. And they took what they knew about DNA from all these other scientists, including Rosalind Franklin's DNA X-ray diffraction, and found that what DNA should look like. They figured out the A pairing with T and the C pairing with G, and that it was in a double helix, and that was the structure of DNA. So they're credited with coming up with what the structure of DNA looked like. From that, they were able to theorize how they think DNA would copy itself, which is still holds true today. Everything that they came up with, including their theory on DNA copying itself, still holds true. It'll happen. So Maurice Wilkins, James Watson, and Francis Crick all won the Nobel Prize in 1962 for their discoveries concerning molecular structure of nucleic acids and its significance for information transfer in living material. You'll notice that Rosalind Franklin's name was not listed. It was not because she was the only gal in the group. It was because they do not award the Nobel Prize to people that have passed away. And by 1962, Rosalind Franklin had passed away due to her research. She was using a lot of x-rays and we didn't know how dangerous x-rays were back then. So she had given herself some cancer from the x-ray radiation and had passed away by the time the Nobel Prize was awarded.